Hello and welcome. In our lesson today, we are going to tackle 4 different application questions on nutrition. So stay tuned. On to the first question. The illustration below represents a process that occurs in the alimentary canal of humans. So the alimentary canal is simply the pathway through which food enters our body and leaves once digestion is complete. It's composed of several organs. We have the mouth, the esophagus, the stomach, the small intestine, the large intestine, the rectum, the anus, and so on. Now, this process can occur in any of the parts that I've mentioned, so it's up to us to determine where it happens. Now, if we look at the diagram, we have a certain compound labeled Q. Now, Q is undergoing a process P in the presence of water. So what happens in this process? It breaks down into 2 constituent compounds, and these are compound R and S. Now, from the diagram, we are given 2 hints as to what this process could be. Hint number 1 is the presence of water. So whatever this process is, it cannot happen unless water is present. And essentially what is happening is that we are having compound Q being broken down in the presence of water to form compound RNS. Now another hint is the presence of enzyme sucrase. Now enzyme sucrase is the one that is catalyzing this breakdown. Now just a reminder, enzymes are substrate specific. This means that a particular enzyme will only work on a certain substrate and no other. So when we talk about a substrate, this is simply a substance on which the enzyme acts on. In this case, if we're having enzyme sucrase, then the substrate will definitely be sucrose. Again, just a reminder, some enzymes take their name from the substrate on which they act on. So enzyme sucrase breaks down sucrose. Now, sucrose is a disaccharide and it's made up of 2 different types of monosaccharides, glucose and fructose. And there we have it. So we've managed to identify what compound R and S are. So either one of these is glucose and the other is fructose. What about this process? Now, there's a certain process whereby a complex molecule is broken down in the presence of water to form simple molecules. This process is known as hydrolysis, and therefore process P is hydrolysis. At this point, we have managed to identify compounds Q, R, and S and process P. So let's move on to part C. Suggest a part of the alimentary canal where the above process occurs. So this process occurs in the ileum. Why do I say so? Because in the ileum, you have intestinal juice, which is also known as succus entericus, and within this intestinal juice, there's enzyme sucrase, which acts on sucrose. So the part of the alimentary canal is the ileum. D, what is the process that is opposite to the process mentioned in B above? So this question is simply asking you, what is the opposite of hydrolysis? Now, hydrolysis, just a reminder, involves splitting complex molecules in the presence of water to form simple molecules. If we were to take simple molecules, join them together to give us a complex molecules, that will be the reverse process. And this process is known as condensation. So you're going to take, for example, glucose plus fructose, join them together to give you sucrose with the release of water. And there we have it. Okay, let's proceed to the next question. The following setup was assembled by Form 3 students of a particular school to demonstrate 2 physiological processes. The setup was initially kept in a dark room for a few hours and then exposed to bright light for another few hours. Now let's look at our setup. We have a beaker containing a water plant, the Elodia plant, and we have a certain gas being produced and collected. Now, the apparatus that were used are made of glass, so that means they are transparent and light can pass through. Let's look at the questions. Part A, what was the aim of the experiment? Now, we can clearly see that if we are collecting a gas, then it's to investigate the gas that is being produced. But which process is responsible for the production of this gas? Now, let us go back to the introduction of the question. We are told that the setup was initially kept in a dark room for a few hours and then exposed to bright light for another few hours. Now, plants in the presence of light carry out photosynthesis, which produces oxygen gas as a byproduct. In the absence of light, in the dark, for example, they carry out respiration. Respiration produces carbon dioxide as a waste product. So both processes are going to happen to the plant at different times. So in the dark, it's actually going to be respiration taking place. And in the presence of light, it's photosynthesis taking place. So we'll be investigating the gas that is being produced during respiration and photosynthesis. Part B. Identify the gas collected when the setup was kept in a dark room. Now, in the absence of light in a dark room, what will happen is respiration. Respiration will produce carbon dioxide. What about Roman 2? When the setup was exposed to sunlight, photosynthesis will take place, and photosynthesis produces oxygen gas. Part C. Why was a submerged water plant more suitable for this experiment? Now, the reason is because a water plant such as the Elodia is adapted to extraction of oxygen and carbon dioxide dissolved in the water. Now, if it were a terrestrial plant, it wouldn't be capable of doing this, and therefore the two processes will not take place. Moving on, what was the purpose of adding sodium hydrogen carbonate to the water? Now, sodium hydrogen carbonate dissolves in water with the production of carbon dioxide gas. Now, carbon dioxide gas, of course, is essential for photosynthesis to take place. So the sodium hydrogen carbonate supplies additional carbon dioxide in order to speed up the rate of photosynthesis. F. What environmental factor needs to be present in both instances in order for the two processes to occur? Now, this is optimum temperature. Optimum temperature is required for both photosynthesis and respiration. And the reason is because these two processes are enzyme controlled. They are controlled by enzymes. And enzymes, of course, are sensitive to changes in temperature. So if you want a fast reaction, optimum temperature needs to be present. Moving on to the last one, suggest 3 ways of increasing the rate of production of the gas produced when the setup is exposed to sunlight. So essentially the question is just asking us, how do we speed up the process of photosynthesis? Number 1, maintaining optimum temperature. Point number 2, increasing the light intensity. And last one, increasing the concentration of dissolved carbon dioxide. And there we have it. I truly hope you're keeping pace with me and that whatever it is we are discussing is well understood. We have one more question to tackle before we are done. Let us now proceed to our last question. The chart below shows a summary of the process of photosynthesis. So we have a chart summarizing the process of photosynthesis or what is involved. Now, photosynthesis is a process whereby green plants manufacture complex food substances from simple molecules. Now, they do this because they have the ability to trap light from the sun, which they then use as a source of energy. Photosynthesis occurs in 2 main stages. The first stage is the light stage. The second stage is the dark stage. Now, the main process that occurs in the light stage is photolysis. Photolysis is a process whereby water molecules are split in order to form oxygen gas and hydrogen atoms. So you have the literal breakdown of water molecules using light energy in order to form hydrogen atoms. Now the hydrogen atoms that are produced will then proceed to the next stage, which is the dark stage. Oxygen gas is given off as a byproduct. Photolysis to take place, water and light should be present. Now it's obvious why water needs to be present, but light also needs to be present because it provides the energy for the splitting of water molecules. Now light, of course, is trapped by the chlorophyll molecules and then converted into chemical energy for use. That is enough prep. I believe we are now ready to answer the questions. Part A, state the requirements for the first stage of this process. So that is the light stage. So we have two requirements, water molecules and light. Part B, from the above chart, what is the origin of oxygen gas that is given out by plants during photosynthesis? The oxygen gas, of course, is obtained from photolysis of water molecules. C, set the role of chlorophyll molecules in the above process. Chlorophyll molecules trap light energy, which is then used for splitting water molecules into hydrogen ions and oxygen gas. Now, another function is that they trap light energy, which is then converted to chemical energy for storage in the adenosine triphosphate molecule. And that, ladies and gentlemen, brings us to the end of our lesson today. I hope it's been a productive one for you. See you next time.